So there have been several very large projects completed over the last 4 years or so that have involved comprehensive sequencing, both DNA and RNA. And what we've recognized as a consequence of these studies is that bladder cancers can be divided into molecular subtypes. The subtypes actually resemble molecular subtypes that we've seen in breast cancers and other solid tumors, which is a big surprise to us. And in particular, one of the subtypes that we call basal, or basal-like, is associated with poor prognoses in the absence of chemotherapy. But as is true of their partners in breast and probably pancreatic cancer and maybe other malignancies, chemotherapy has major impact on survival in those patients. So one concrete thing that's happened as a consequence of genomics is that we've recognized the clinical significance of these basal cancers and how they are probably aggressive at baseline but need to be managed with chemotherapy. In parallel, the DNA sequencing studies have identified DNA repair mutations that are associated with response to neoadjuvant chemotherapy. And so now there are a series of prospective studies planned to determine whether or not the presence of these DNA repair mutations is sufficient to predict complete or near complete pathological response to chemotherapy, which in turn would mean that patients with these mutations might be treated with neoadjuvant chemotherapy and not cystectomy, as they're currently managed now. So a bladder preservation approach. And then finally, there are targets in these tumors, some of which we were already aware of, like FGF receptor 3, others that we weren't. And those targets provide us with new opportunities for therapeutic intervention.